thanks for inviting me. Good to be back in a way. I know many of you, and I really miss coming to your country, and I really hope I can do that soon as the skies open.
Hopefully, yeah.
Yes. I'm going to present a bit on DSC, calorimetry today, regarding how it's used in a pharmaceutical environment, and also the background, some of the application, and a bit of stories I've been seeing through the years. And within calorimetry, yes, there are two major disciplines, two techniques. So one is isothermal titration calorimetry, where you monitor an interaction. So component A binding to component B, and monitoring the heat that is being evolved upon an association. We get affinities and enthalpies and the thermodynamics of that interaction. So that is done at a set experimental temperature, which we program. And we have then a differential scanning calorimetry, which is a thermal shift assay, where we heat up a sample, a biological sample, and monitor how much energy is required in order to denature or destroy that structure. So it gives a fingerprint and a stability indicator, or a predictive method in order to monitor stability and such. So just one slide on ITC, which many of you might recognize, is that we have a ligand in a titration syringe. We have our macromolecule or receiving molecule in the sample cell, and we program the titration syringe to deliver a certain number of injections of a certain volumes. That's done automatically. We monitor the heat, and then we derive different variables. So enthalpies, affinities, stoichiometry, which is a critical aspect when looking at sample activity. A big field that is growing more and more is kinetic reactions, so enzyme kinetics, inhibitors, and CMC measurements. So it's one of those, it's an old technology, and it will really deliver good data if you treat it nicely. So moving on to the DSC. So the design of DSC is that you have a sample cell or a capillary and a reference capillary, which in the sample cell you will fill it with your biomolecule, being it a protein or a liposome or something with a structure. And in the reference you will place your buffer. And then the instrument will apply a thermal gradient and heat the whole core according to that gradient. And when it reaches a certain critical threshold, you will start to denature your sample, and it will then need additional heat. The cell materials are tantalloid, which are extremely inert and resistant, because we're kind of cooking eggs in a very small compartment, and these are non-removable cells or capillaries. So we really need to be able to use harsh cleaning conditions like harsh acids or bases. So just a cartoon on a DSC experiment. So we have the sample cell, and sandwiched in between is a series of Peltier devices. And a Peltier device can either be used as actively or passively. So you can heat it or use it as a sensor. And in this case, the one that are in between the two measuring compartments is acting like a sensor. So if there's a temperature difference or a heat flow detected inside the Peltier device, there will be a voltage that's been generated, and then we can compensate by applying additional heat to one of the sides in order to make sure that both sides have the same gradient. So we program the instrument, we press start, and during the heating phase here, there's not much happening. Both cells are following the gradient. At a certain point, you reach that threshold, your sample will start to denature, and you're taking up to higher energy level, and it will start to absorb heat. So it will actually take heat from the core, creating an offset in the Peltier device, and that will generate a voltage output. And the instrument response here is to apply additional heat to the sample cell in order to maintain a zero voltage output. And this is done throughout the full denaturation profile up to the point where we have a fully denatured state in the sample cell and we have reached our final temperature. So the parameters we derive from a DSC, of course, we have the TM, the thermal transition midpoint. And also the area, the calorimetric and the Van 't Hoff's area, and delta CP values. So all these parameters are very characteristic for a specific sample type, experimental conditions, buffer conditions. So we can really get very high resolution information from a DSC scan. So for instance, delta CP values, the larger those are, the higher difference there is in accessible surface area from a native to a denatured state. If your Van 't Hoff's enthalpy is very high, meaning that you have a very narrow peak, that means that you have a high cooperativity of your unfolding. Vice versa, if it's a very wide peak, then you have a wide structural distribution of your sample. And you can observe several domain stabilities. So DSC is one of the prime characterization tools for monoclonal antibodies. And I have some examples of virus and AAV studies as well we're going to look at. So it's a direct measurement, and most samples will give a trace, but we are reliant on having some sort of structure. So if you want to run your proteins at pH 2, where everything is kind of denatured, then you won't see anything. You need that structure content. And which concentrations we recommend? Well, it all depends on the molecular weight and the structural content. But a good starting point is somewhere around 0.5 to 1 milligrams per milliliter. And we need approximately 325 microliters to do one experiment. What is unique in terms of thermal shift and technology is that it's a quantifiable thermal shift assay. So the higher concentration you have, the higher signal you will have. And on the right, this is 3 different samples, or the same sample, but run at 3 different concentrations. And the normalized data then overlays because you have the same kind of heats and TMs coming out of these data. So it's used for fingerprinting. I have some slides on the similarity assessments of originator and new products and such. This is one of a very nice example of how the area itself can be used. This is some work I did for a crystallography department in Oxford, where they were having problems with their getting crystals of a certain protein. And we tried different formulations, so different lipids and such, 5 different conditions. We prepared 5 different samples and we put it in an automated system where we kind of introduced a time delays to let the second set of samples be incubated in those specific conditions. So on the top right, you see the conditions, the original conditions. So the big peak here is the protein fresh off the column. So a nice big peak, but after 24 hours in that specific buffer, the peak area has been significantly reduced. So you're losing structure, it's dying in that formulation. And we found that this C12E8 formulation, which is a commercial lipid, I think, maintains the same area and also gives a higher TM. And this is what they then selected to move forward in their crystallization and production process. And this can be the same. This is just an example how DSC is used, which kind of formulations, which kind of additives or pHs one can do. Also, one good example here is with combinations. So running the samples individually of different excipients. So is it glucose, or should it be methanol or ethanol? Does it increase or decrease the stability? And getting that final combination where we have an increase in TMs and profiles, rendering a longer shelf life, for instance. This is how a conventional monoclonal antibody screen would look like. So these are different variants of the same antibody, humanized and unhumanized versions, different buffers, and so We see, this is also the first time we see the high data resolution we can obtain with DSC, where we see 3 different domains accredited to a CH2 FC domain and then the CH3 domain. And already here in an industrial environment, we have a good data point where to perform accelerated stability assays. When we do these stress tests, one needs to be able to motivate why do I actually use it to stress the data, my samples at 60 or 40 or 70 for 2 weeks. So if you do your stress test a bit further away from your onset temperatures, you will have less data spread. But if you are just at your onset, you will have higher differences between those who can tolerate it and those who cannot. That's because the aggregation process and precipitation process is a cooperative effect. So you get better data spreading of your data. And also DSC data supporting these stability assays is an FDA approved method as well. So that will be that's a good benefit. We can deconvolute domains, see how different domains shift depending on different modifications or scan rates or buffer compositions, identifying weak links. So where should I target my chemical modifications on, and so on. So deconvolution is a bit dangerous as well, because model fitting in thermodynamics is only valid for reversible systems, but by using high enough scan rates, one can, to a certain degree, quench the aggregation kinetics to get the characterization, feature more dominant. And vice versa. If I'm looking at long-term stability assays, then I want to use a very slow scan rate, because then I'm more interested in the aggregation kinetics. How does the aggregation actually pull out my sample from solution and aggregates it and precipitates? So different kind of, a very wide range of applications available.
Peter?
Yes.
We have a question here.
Okay.
Will you be kind and to discuss studying of quality attributes of materials in LT DSC?
In what?
LT DSC.
In low temperature DSC. We don't do low temperature DSC. So this goes, these samples needs to be in solution. So the lowest temperature we can do in our systems is from 2 degrees, and the temperature range goes up to 140 degrees. The majority of calorimeters in general, ITCs and DSCs, I would say that 95% are industrial range. So this is where you look at wetting of a powder or melting of a solid or phase transitions and such. And there are many different companies doing that. So PerkinElmer or Cetaram or any of these. The microcal range is for biological active samples, so for in-solution samples. So that's also why we have fixed in place capillaries and cells in order to maximize sensitivity. So low temperature, that would be sub-zero applications, is not really that something we can do with our current DSCs.
Thank you. Yossi, if you have any question regarding this, you can write me, and I will actually can unmute you if you want to speak with Peter. Okay, just let me know.
We're good?
Back to you.
Back to me. Okay, cool. So here is look at an example of how fingerprinting and that there's additional information. Not only the TMs are important when we look at overall stability and sample properties. So this is a immunoglobulin, for instance, looking at the effect of polysorbate 20 or tween 20. So these are from this is published data where they investigated how concentration series would influence the stability and profile of immunoglobulin. And they were looking at the main transition TMs only. So, and looking at the data now as such, we don't really see a difference. So they have the same TMs of these two peaks, and they could conclude that the presence of tween doesn't actually influence the stability or properties of the sample. But revisiting the data, looking more in detail, for instance, looking at when does the peak actually start, when does the process start, and how wide is the peak, looking at the Bandt-Hof's enthalpy or half peak widths at half area. There's a different story evolving where we see that with increasing concentration of tween, you would have a later onset. So it's more stable, and also your peak becomes narrower. And that means that you have a more homogeneous population of your sample. And that correlates to that if the full population is in the sample have the same conformation, they are equally affected by the heating process. So they will unfold at the same time, resulting in a more narrow and higher intensity peak. So a homogeneous population, that's a very good thing because that is much more predictable for long-term processing and later on in the development stages, for instance. So getting these information. So there was a significant influence in the presence of these additives, but not seen in TM only. And one of a good example I did for a university in Germany, where we looked at binding, and this was coming actually from an ITC experiment where we did some tests, and we could confirm that the FL protein, I don't know which protein this is, binds to a ligand B. So that was confirmed. And then we had some samples left, and we put it in the DSC. So the black trace is the protein alone, and that displays a distinct two-domain protein. And when we incubate it with a ligand, we see a TM shift of the first domain, so to a higher stability. And this is your binding domain we were observing. Whereas the second domain, and this can be, this is an inactive part of the sample in terms of binding. So you can do very cool stuff with interaction assays as well, or screenings and such. One more example of binding is, again, this is a bit more traditional. So this is a phenylalanine binding protein from a human version, where we are looking at an increasing concentration of phenylalanine. We get a full shift of the profile at a higher temperature. So that is what to be expected. Different story is seen if we take the same protein from C. elegans, is that instead of having a TM shift, we're seeing an evolution of a structure. So in the first shoulder there with zero phenylalanine, the more phenylalanine we add, higher how the structural content that first part of your sample will have. So again, high resolution interaction data can be obtained with a fairly straightforward titration series as such. And this is a liposome example, just to showcase that the experimental or application range is very wide. This is a liposomal adjuvant in vaccine formulation, where the gray trace is the first heating ramp, and then there's a cooling of the sample, and then we do a rescan. And this was kind of tricky because in the first trace, what we see is that at 30 degrees, we have a pre-transition. So this is where your lipids are kind of snugging together in the membrane. Then you have a fully demycelization process. And then at 45, it's a bit unclear what that is, but I have some good references from Chezi that it might be the fatty acid release. Whereas in the second or a heating process of the same sample, the pre-transition is not longer observed, and nor is the fatty acid content release. The correlation, these are our commercial and active samples, and what they could say, and this was in Denmark, was that the lower degree of that transition they see at 45, that presumed fatty acid release, the higher long-term or longer long-term stability they could observe in their direct samples. So they were aiming to minimize these features at 45 by using different combinations and production techniques and so on. Now looking at virus-based products, this is very popular nowadays, so to say, and it's been around for quite a while. So characterizing different virus-like particles, full influenza viruses, all sorts of different. The virus capsids are very well suited. They have good stability. The productions are you can get quite high quantities and so on. So there is a lot of literature and history in characterizing virus-based products. And something that is of course more and more present is this adeno-associated virus for gene therapy. So using viruses as delivery of gene modifications or viral vaccine production and such. And this is just out actually. This article is available in open access. Was yesterday. I'm just going to highlight the DSC part, but in this publication, STECMALs, DLS, and different kind of biophysical techniques was used in order to characterize how AAVs could be qualified, or what kind of serotype they were having, the degree of loading of the payload, and so on. So the TM indicator of AAV serotype, but it depends on all sorts of factors. So what kind of heating rates we're using, or sample concentrations, and unfortunately, TM is generally quite similar both for empty or full capsids of the same serotype, and there's no direct correlation of TM and the dynamics and when does it actually release its payload. And TMs from DSC and DSF could capture the disassembly of the entire capsid. So that's a big thermal event or thermodynamic event, of course. But maybe more important, the temperature of viral DNA release, TD, that is more important for the actual functionality of these AAV vectors. So there is kind of a balance. The more stable I create my vectors, how will that actually influence the TD aspect? Will it be more difficult to deliver the payload to the preferred tissue or the place in the body where it should be active? This is, of course, there's a huge amounts of research being done, finding out how these dynamics and combinations are actually working together. So here is just one example. What is interesting is that we're looking at and comparing a loaded and an empty RAAV5, so recombinant adeno-associated virus strain 5 with DSC. And there are some differences, subtle differences. So first of all, this is not obvious, but if we zoom in and take a look, so something happens first in the temperature range between 50 and 75. And this is a temperature-induced process. And this is actually when, due to the temperature stress, the payload, the single-stranded DNA, is leaking out of the capsid. So it's a very subtle difference. And what was observed is that with fully loaded AAVs, you could see a baseline drop. So a quite profound kind of difference in relation to an empty AAV sample or the baseline. Secondly, what was observed was that the pre-transition is most pronounced in empty, AAV vectors. And main transition, that's a highly cooperative, and then we're looking at the protein capsid, so they are very similar. And in the loaded capsid, we could see the single-stranded DNA melting occurrence as well at 95 degrees. So looking at these different differences and also in combination with other techniques, it's possible to then identify degree of loading, serotype, storage conditions, and such. A couple of years back, there was a kind of a review of technologies used for in the biopharmaceutical area, ranging from candidate selection, biosimilarity, and such, where they compare different techniques. And this is an independent study where they found that actually the DSC application is very useful in all of these processes. So the more bright that this box is, the higher usefulness, so to say, specific region. So some technologies are really good at something but lack the capability in others, whereas DSC has a fairly constant kind of bright or useful application range. And it's more sensitive than one might think, because here is an example of, for instance, oxidation of methionine, for instance, where the higher degree of oxidation in a CH2 domain, the lower TMs were seen. And this was also seen at the single amino acid chain level as well. So very sensitive and reproducible, looking at the product quality, for instance. And batch release in manufacturing environments. So getting that fingerprint, so different manufacturing sites all over the world nowadays, looking at the reference sample, how does different manufacturing lots actually correspond? Do we have a shift? Did we change a chemical supplier? So it's a very powerful tool, considering and thanks to that you have a quantifiable signal. So the more structure you have, the higher signal you will have as well. Biosimilarity, and this is also accepted in FDA and regulatory environments, where you see the full profile, looking at the fingerprinting, comparing an originator and a biosimilar product. So not only for batch release, but actually also for confirming that there is no difference between the actual originator and biosimilar product. And in the software, there are inbuilt functions. So for instance, one example is the similarity report, where we look at the residual differences between different traces. So how the practical, what you do is that you select first one data set, which will be your reference, and then you select further one, and then it will compare to the first one. And you have a similarity score that ranges from 1 to 0. So 1 meaning it's absolutely similar, whereas 0 is meaning that it's totally different. So that's also a very quick and easy way of confirming that you have the same sample running over and over again from a production line or in a QC environment. So we have also this 21 CFR Part 11 option as well here, where we can activate audit trails and electronic signatures and even link it together to an access control, limit the degrees of manipulation freedoms different users will have and restrictions, all in order to comply with the regulatory body requirements. Similarity Statistics. This is a tool if you want to further explore to make larger groups. So what you would do here is, for instance, you would run the same sample maybe 10 or 20 times to get the variance within your specific production line. So, and then you can use those values, you know, to put it into a similarity assessment tool. So here, for instance, we can do it, for instance, which is most similar and least similar to a certain set. Also, similarity scores will be reported. You get variance and standard deviation, and 95% confidence interval. And these values will then be transferred manually into the assessment, where you will create a certain a similarity assessment report, which you can reuse over and over again. So you will create your foundation of the similarity assessment by running the same samples over and over again. And then you would have that database, so to say, and then run compare different samples to that similarity assessment. And it will give, for instance, pass or fail criteria based on either your input or the actual similarity statistics. And this can be, of course, calculated or changed as you would like to. And then once you have done your done that, you will then lock it in your report, which we can then use. And here is just how you, an example, how you would change the upper and lower bounds or TMs, or what are the criteria for a pass or fail outcome. The experimental setup in an automated system is it holds 696-volt plates in a temperature-controlled environment. You would program it to do your runs. What is a buffer run? Which is a protein? How to clean it with detergent or with a heating ramp of detergent, the input concentrations, what kind of sample names, and so on. How much water or buffer to rinse it with. You can edit, for instance, you can do rescans of the same sample. And of course, where should the scan start and end, what ranges and such. If you want to do down scans, so heat it up and cool it down. So it gives a wide range of options in operation. What tends to be the situation is that in an environment, you will use maybe 5 or 6 different run types. And of course, you can save all these experimental setups and also analysis methods to tailor your needs. And these analysis settings in the analysis part, you can import them to automatically be deployed when the files have been created. So you can be sure that you use the same, for instance, baseline function or ranges where it should be integrating from, and so on. And this is in order to minimize the manual input or modification of the data. Here's, for instance, a couple of screenshots on the analysis part, where we can select which buffers we should use to subtract, what ranges for analysis. We can import already existing analysis settings, so where the ranges and what kind of baselines and buffers should be subtracted. You can do model fitting or just do TM finding, looking at the raw data. And this is where you would be exporting your analysis method and then be able to deploy it into the actual run file without physically needing or to actually analyze. There's algorithm to find peaks and shoulders. You can also add manually by right-clicking on a data point. This is also useful in order to minimize the manual hands-on work. So defining where your main transitions are. And with that, I would like to thank you very much for your attention, and I'm open for any questions.
Any question, guys? Nothing yet. I just want to thank you very much for joining us and Malvern, always such a great cooperation with you is great for our customers. Thank you.
Thank you.
Actually,